All right, so let me just quickly, for those that haven't taken a look at so here's the announcement I posted yesterday about a lot of the reminders I just told you guys about. And also here we have, resources that I really think you guys should take a look at. And for under the additional lecture resources over here, there's a lot of tables, including this endocrine table. And this endocrine table has the hormones, where it's secreted from the target organ or tissue, and then what are the functions, what are physiological effects of this hormone. And so, you're going to want to spend some time being able to fill this out, help you study the endocrine system. In the lab content overlaps quite a bit with the material you need to know for lecture. All right, so you'll get also an opportunity in lab to study hormones, their function, their effects, which glands they're coming from. So here, for the lab, like the special senses lab, there's a lot of overlap with what you guys do in the lab and what we're talking about. So we had finished up with talking about hearing last time. Did anybody have any questions on that? I think putting things in motion by looking at those animations and videos really helps. I think trying to look at a three-dimensional structure that is on a static two-dimensional flat, projection or a piece of paper is really challenging. And then also trying to understand how this system works, in dynamic fashion is really challenging without putting it in motion. And so that's why I have a variety of animations and videos up there for you guys to go through and see multiple times. And I strongly encourage folks to, go through this whole series of events, sequence of events, with the animation until you're bored out of your mind. Because that's when you really know the material, right? So that's one of the metrics that, I think helps with making sure that you know that material fairly well. And so here with hearing, we looked at the cochlea, and specifically within the cochlea was the organ of Corti, which was involved in the hearing. Now what we're going to do is we're going to move on and take a look at the vestibule and the semicircular canals. The utricle and saculae, which you don't need to know, are part of the vestibule. All right, and so we're going to focus on this other part of the inner ear that's involved with equilibrium. Okay. All right, and so just be sure for each one of the senses that we talked about that you understand the sequence of events that goes on in order to be able to generate that sensation and where it's routed in the brain for being interpreted, identified, and so forth. Okay. So equilibrium, we're focusing on this part of the ear with the semicircular canals and the vestibule. All right, so there's kind of two types of equilibrium. One's called static equilibrium, the other one's dynamic. Static, you can kind of think about as being very linear momentum or linear acceleration. All right. Dynamic, you can think more about angular momentum or angular acceleration. And so those two are being sensed in the semicircular canals and vestibule. So here, when we talk about static equilibrium or linear, we're talking about moving, say, going forward, back, side to side, or up, down. So when you're sitting in a car, and the car accelerates, right? So in that case, that's linear acceleration, and when you stop, that's also deceleration, so it's linear. When you guys go up and down an elevator, that's linear. That's being detected in the vestibules. So if you didn't move your head, and when you sat down, right, that movement, that change in height was being detected by the vestibules of the inner ear. We're involved with this linear detection of equilibrium. Semicircular canals, on the other hand, are involved with angular, so kind of more spinning types of equilibrium. So spinning around, when you guys do your backflips, all right, you do your cartwheels, okay, all right, so all of that is much more triggering the semicircular canals than the static equilibrium that's found being detected by the vestibules. Is that making sense to folks? So here, the linear or static equilibrium is more in a single plane, and then the dynamic is much more circular motion. All right, so with the static equilibrium, what we have are hair cells that are embedded in what's called this autolytic membrane, and sitting on top of the autolytic membrane are these crystals called otoliths that are made up mostly of calcium carbonate. And there's two orientations in the vestibule of this structure. One is situated so that the hair cells are pointing out, and the autolytic membrane is sitting here. The other orientation is where the hair cells are pointing up, and the autolytic membrane is sitting on top of that. Okay? So here you can see that this one that is oriented so that the hair cells are more horizontal pointing, they're going to be able to detect motions that are up-down. The other one, where the hair cells are kind of sticking up and the autolytic membrane is sitting on top of them, are going to be detecting motions when you are like accelerating or decelerating a car or moving side to side. Okay? Now, how this whole system works is that these are our mechanoreceptors. And when the hairs move, they are going to trigger a signal along this these neurons that's going to be detected as a change in equilibrium. Now, how do these hair cells move? Well, this fluid kind of has a weight associated with it because of those otoliths. So think about having like a bucket of water, right? So if you accelerate really quickly, what happens to that water? The water kind of washes back a bit, right? Okay? So the water doesn't move at the same pace as the bucket and the rest of you, right? Okay? That's something happening here with these otoliths. So when you go up an elevator, right? You go up, so this part is going up, but this part lags behind, meaning it tugs a little bit on these hair cells. And that's the mechanical stimulus that's being sensed. Right? So it kind of sloshes around a bit. Now the same thing happens if you, say, step side to side with a bucket of water, right? It's also going to, that water lags behind a little bit, just like the otoliths in this autolytic membrane. Lags behind a little bit so that, it causes these hair cells to be mechanically stimulated to send a signal. Is that making sense to folks, how the system works? And so the examples of kind of accelerating, decelerating in a car, and also going up and down an elevator, are much more acting upon the vestibule than the semicircular canals. The example I think a lot of the books give of where someone is nodding their head, you're actually invoking a lot of the semicircular canals in this system, and not just the vestibules. All right, so here, even though it's giving an example of how, these otoliths and autolytic membrane, these hair cells function when you move your head, nodding, up, down, and side to side. It's not the only part of the equilibrium system being activated in this type of motion. All right, and so here, these are really involved with changes in gravity. Right? So here we're really using gravity to help, move the hair cells by this otolith and autolytic membrane combination. So is that clear to folks how this system works? So usually both the vestibule and the semicircular canal system get activated in some manner. So this is linear or static equilibrium. Dynamic equilibrium, like I said, is about, kind of rotational or spinning. So here, nodding your head is going to be part of this system. All right. So semicircular canals, they're filled with fluid, just like the vestibule. And they're oriented in 3 planes. And I got this deluxe version of this model to show you guys of a semicircular canal. It's deluxe because it has different colors. Right? All right, so the semicircular canals, we have one that's going in this direction, another one in this direction, and another one that is going in this direction, which means that if we take a look at our head, so if we spinning around, that's going to move fluid in this semicircular canal, and that's going to be detected by structures that are at the base here. That's called the ampulla, crista ampullaris, has this cupula, and because it has a fluid in it, that fluid, just like with the bucket of water, okay, the fluid doesn't exactly move as fast as the canals do, right? And so here, that fluid is going to cause these hair cells to be mechanically stimulated, and you're going to detect that motion, all right? So here, if we rotate our head side to side like that, and then up, down, right? That's going to be detected in this semicircular canal, right? And then nodding like this is detected in this semicircular canal. So we have 3 planes of motion basically that are we can detect because the semicircular canals are in those 3 planes. And each one at its base has a detection kind of organ, microorgan, this crista ampullaris. All right, so to kind of show this, I'm going to show you guys a video. right. Did that make sense to folks? Kind of how the system functions? So it has to do with the inertia of the fluid in the semicircular canals. And so what happens if you spin around for a while and then suddenly stop? So what does that feel like? The room keeps spinning, right? So why is that? Why do you guys think that? The fluid is still moving, right? So just like when you think about the bucket of water there, you're swinging the bucket of water around and then you suddenly stop. What happens to that water? It kind of sloshes around, right? Same thing is happening here in the semicircular canals. Is that making sense to folks? All right. All right, so head position and balance equilibrium is part of part of equilibrium. Remember that we're moving endolymph and it's triggering the hair cells, which are really mechanoreceptors on the hair cells. And here, rotational or spinning is semicircular canals, and the vestibules involved with kind of this linear motion. So forward, back, side to side, up, down. All right, and then so this is really important for gravitational changes. And here we're sending these signals along cranial nerve number 8, which has both information coming in from the cochlea as well as these equilibrium parts of the inner ear, right? The vestibules and semicircular canals, right? So now you can understand why it's called vestibulocochlear. The cochlear part is dealing with hearing. The vestibulo is referring to the equilibrium or balance portion of that information. All this is housed within the inner ear, which is all within the temporal bone. So if you go slightly behind your ear, there's kind of a bony protrusion there, and that's where a lot of these structures are located, right there. All right, so where do we process all this? Well, it gets funneled into our brains along with a lot of other information in order to get some inkling of what equilibrium is for our body. So we get cranial nerve number 8, which is the vestibulocochlear information. And then we also have proprioceptors. Remember, those are detecting stretch of tendons and also muscles. So we have the Golgi tendon organ and then also muscle spindles that are inside of muscle fibers detecting stretch. And then we also have visual input coming in, right? So if you're taking a look at a room and it's slanted, right, you're going to input that in order to maintain equilibrium, in order to walk from one side of that room to another. That input's going to be taken into account. And the parts of the brain that are really important for this, so part of it's in the insula, which remember is going to be this deeper part in between the lateral sulcus, and so that is involved with processing a lot of the vestibular information. And then for a lot of the other information that's going to be going into the cerebellum for processing. So a lot of the information on postural reflexes, and it's taking into account inputs from other parts of the brain. So like I said about the slanted room, if you're going to have the intent to walk across, a room that appears slanted, the intent starts off, that plan to do that starts off in the motor association area, then that plan is sent to the primary motor cortex, which is going to send that out. And part of that plan that gets sent out is incorporating the information from the visual input and also the proprioceptors saying, all right, how much have I stretched this tendon, this muscle in order to maintain equilibrium? All that is factored in to this process of, say, walking, from one end of the room to another. All right? Does that make sense? All the things that come in together in order to maintain equilibrium here. right, so the last thing that, I think a lot of people are always interested in, what about motion sickness? Motion sickness, the best idea out there for what causes it is that there's a conflict between sensory information. So if you're sitting in a room on a boat that has no windows, and your inner ear is detecting motion side to side, front to back, rolling, and all of that, but your eyeballs are not seeing the room you're in doing any of that, right? There's a conflict of information. And so the best idea out there for what, is going on is that there is this kind of, disagreement among the senses, and your brain's trying to make sense of that. A lot of motion sickness medications, the thought is for how they work is that they kind of tamp down a lot of the processing to try to minimize, this conflict that happens is the best way to describe it. All right? Does that make sense to folks? All right. Okay, so that's the end of the special senses. So why don't we take a 5-minute break, and then after the break, we'll talk about the endocrine system. All right, so I think we'll go ahead and continue on. We're going to switch gears now. So we're going to switch into talking about the endocrine system. Before we do that, I just want to kind of recap a little bit about the special senses when we talk about receptors. Right, so which sense has, say, chemoreceptors or senses? So which senses or sense has chemoreceptors? Olfactory, absolutely. What's another one? Taste, gustation, right? Those are the two with chemoreceptors. And then what type of receptors are involved with vision? So photoreceptors, right? So chemoreceptors, photoreceptors, and then when we talk about hearing in an equilibrium, what type of receptors are we talking about there? What's the stimulus that activates? Mechanoreceptors, right? So we got chemoreceptors, photoreceptors, and then also mechanoreceptors, right? Okay. All right, so for the endocrine system, hopefully you guys are going to see some similarities here between the endocrine system and synaptic or neurotransmission, where we had communication between a presynaptic and a postsynaptic cell. All right? Let's start off with talking a little bit about some basics. So here, if I have some sort of water-soluble chemical messenger here. Right? So I got a water-soluble chemical messenger. I'll draw two of them. All right? Based on what you know about the cell membrane, if they're water-soluble, can they easily cross to inside of the cell? No. So if these chemical messengers are going to relay communication from outside a cell to inside a cell, where does the receptor have to be here? Can it be inside of the cell? No. It's got to be, so it's got to be pointing outside of the cell, which means that it's embedded in the membrane, pointing outside of the cell. This is something we saw a lot with neurotransmitters in synaptic transmission. So here, okay, for water-soluble chemical messengers, okay, when we get into the endocrine system, we call these guys hormones, and we'll talk about why that is in just a second. The water-soluble ones, their receptor in their effector cell is going to be membrane-bound, which means that when one of these chemical messengers binds to the receptor, just like what you guys saw with G-protein coupled receptors, it causes a series of things to happen within the cell, such as genes being turned on and off, proteins being turned on and off, enzymes specifically, right? Okay. Does that make sense to folks? Hopefully this jogs your memory a little bit of what you saw with synaptic transmission. Now, the other type of chemical messenger that we have, instead of being water-soluble, is going to be lipid or fat soluble. Based upon what you guys know about the cell membrane, if it's if it's fat or lipid soluble, does it need help to get across that cell membrane? No, it can get across fairly easily. Does a receptor have to be membrane bound for these guys? No. It can be actually within the cell itself. And that's what we're going to see with some of these hormones that are lipid soluble. The actual hormone receptor is within the cell. And so here, these guys can get across the cell membrane and then bind to the receptor, which is within the cell, so we say it's an intracellular receptor. Right? When it binds to this receptor, then you're going to get genes turned on and off primarily, but also in some cases proteins are going to be affected as well. Okay? So here we kind of have two broad classes. We've got water soluble and lipid soluble. Each one of those, you can tell where the receptor is going to be for them based upon this chemical characteristic. Any questions on that? It's clear? Okay. Next thing we're going to talk about is some terminology. So, has everybody got this? I'm going to erase it. Yeah, okay. All right, so there's kind of three terms we're going to explore here. One is autocrine, or autocrine mechanism of signaling. When you hear the word or the prefix auto, what is that? Self, right? So here what we're talking about is if we have a cell, we've got a cell, and it generates some chemical messengers. All right, so it's going to secrete some chemical messengers. In order for a cell to respond to these chemical messengers, what does it have to have? Receptor, exactly. Absolutely. A receptor. And so here, with autocrine, the cell that secretes the chemical messenger also has the receptor for that chemical messenger. Which means that it stimulates itself. Right? Autocrine mechanism. Okay? That the cell that secretes that chemical messenger also has a receptor for that chemical messenger, meaning that it can respond to it. So this is an autocrine signaling or autocrine mechanism of stimulation. Paracrine So if we have a cell that secretes some sort of chemical messenger, paracrine means that cells nearby in the vicinity of the cell have the receptor. So not the same cell, but cells nearby. They have the receptor. And when this chemical messenger binds to that receptor, so when that chemical messenger binds to this receptor, these cells are affected. So they're going to have a response. Okay? Where genes get turned on and off, and then also we get proteins being turned on and off. So far, from what I've drawn, would you say these chemical messengers are water or lipid soluble? Water soluble. So what makes you guys say that? They all have receptors, but where is the receptor? Yeah, so it's not outside a cell, it's on the membrane. It's membrane bound. So it's facing outside a cell, but it itself is not just floating off in some sort of, kind of weird space out in the cell, out in the extracellular fluid. It's membrane bound and outward facing. Okay? So be sure to know that distinction there. It's membrane bound. All right, hormones. We have a cell that makes a chemical messenger. Secretes that chemical messenger. The cell secretes that chemical messenger into the circulatory system, blood vessels, capillaries specifically. Right? Now, if it secretes that chemical messenger into the circulatory system, where does it go? Everywhere, systemically distributed, right? So it's going everywhere. So if we have, say, a cell down here that has this receptor on it, is it going to respond to this hormone? It doesn't have the right receptor. So what cells are going to be able to respond to this hormone? Cells that have the specific receptor for that hormone, right? So in this case, I haven't drawn it yet, okay? But we'll stick with these being lipid soluble, or sorry, not lipid soluble, water soluble chemical messengers. So if we have a cell that has this receptor for this, it's going to be able to respond to it. Chemical messengers that are released in the bloodstream that get systemically distributed, we call hormones. Right? Now, which one of these three examples reminds you of neurotransmitters? What's that? I thought paracrine? Yeah, absolutely. Paracrine, right? So here, paracrine, so where we have presynaptic cell, maybe a postsynaptic cell, and this would be our synapse, right? Okay? So neurotransmitters are examples of a paracrine mechanism of signaling. So here, if we're talking about the neuromuscular junction, right, what cell is this? Well, let's start off with what neurotransmitter is this? Acetylcholine. What cell is this? It's a presynaptic cell, and in the neuromuscular junction, we call that the motor neuron, right? And then what was the effector? What cells had that acetylcholine receptor? Skeletal muscle, right? So here, paracrine example is going to be neurotransmitters, what we saw. Here, we haven't seen too many examples of this. Turns out the immune system has a lot of signaling molecules that it sends out. In a lot of cases, the same cell that secretes that chemical also has receptors for it. So some T cells activate themselves through this mechanism. Now, the other thing is that it doesn't mean that because this chemical messenger can activate the cell that secreted, it can't also activate another cell in the nearby area, right? So it turns out that, here we're just talking about mechanisms. It doesn't mean that the chemical messenger is only doing one mechanism. It can blend those mechanisms together, right? So if I had another T cell that was sitting over here that had the receptor, for instance, that was for this chemical messenger being secreted from this T cell, it could be activated, right? And if the secretion's coming from a different cell, then we would say that's paracrine. So it's just a way to describe the activation mechanism. Everybody with me on that? Now, is everybody clear about hormones? Because this is really important, because a lot of hormones are released into the bloodstream. And do all cells respond to all hormones? No. Only the cells that have the receptor are going to be able to respond to that specific hormone. All right?
So how directly does the hormone feel?
Oh, yeah. So when you get the cardiovascular section, we'll talk more about it. Turns out, the capillaries are a little bit leakier in endocrine structures. So if you remember back to when we talked about histology in tissues, there's one type of tissue called reticular tissue, and all we said is that's found in a lot of endocrine glands, endocrine structures. And if you remember that reticular tissue is also had a lot of space, and one of the reasons is because there's kind of leakiness with the blood vessels, or specifically capillaries. And so that's really important, and we'll talk more about that when we get to cardiovascular section. And areas that need access to the circulatory system, the blood supply, right? Either for monitoring substances in the blood supply or secreting things into the blood or taking things out of the blood. Those capillaries are going to be leakier. They're going to have more holes in that area so that those surrounding tissues can get access to the blood. Okay? Yeah. Yep. So gas exchange, absolutely. Yep. Esme? Oh, blood vessel. Yep. Got it. Any other questions? Does this make sense to folks? All right. So water soluble, lipid soluble makes sense. These mechanisms here. So if a cell responds to a hormone, it's got to have that receptor for that hormone. If it doesn't have a functional receptor, notice I said the word functional, okay, it's going to have problems, right? And that can cause major problems systemically. All right. Oops. All right, so we kind of talked about these already. Hormones are generally released from particular tissues called glands, and the hormone targets and functions are something we're going to talk about a lot of. We talked about water versus fat or lipid soluble already. We talked about also using what we know, right, with neurotransmission, synaptic transmission there, right? And so here, some of the major glands, you guys are going to see these in the lab. So hypothalamus and pituitary. You're going to find out why the pituitary is called the master gland. It has a lot of hormones that regulate other hormones. And the hypothalamus is intimately connected there. And then we have the thyroid gland, and embedded in that thyroid tissue is specialized tissue that makes up the parathyroid, or like little buttons inside the thyroid gland. We also have the adrenal gland that's sitting on top of the kidney. And then we have the pancreas the pancreas has an endocrine function and also something called an exocrine function. So if something's endocrine, it means endo means within, right? So secreting substances, chemical messengers that are staying within, say, our tissue system. Exocrine, so if you remember exocrine secretions, what was an example of that? No, not insulin, because that's going into the blood. So it's staying within our tissues. Exocrine. So think back to the skin. Sweat. Yeah, sweat's an exocrine secretion, right? Sweat glands, they're secreting something that's going to be deposited kind of outside of the body. And remember, there's structures that are connected to the outside of the body, right? Like the GI tract. So any secretions going into the GI tract, the intestines, small intestines, those are considered exocrine secretions. Everybody following me on this? So all those digestive enzymes that help break down things within our intestines are called exocrine. We'll find out when we get to the digestive section that pancreas also is involved with those exocrine digestive secretions for breaking things down. So there's an endocrine and an exocrine function to the pancreas. All right? Okay. Yeah, you probably can consider amylase an exocrine secretion. It's part of saliva. All right. Endocrine versus exocrine, we talked about that. Hormone and neurotransmitter, do you guys understand the difference between those two from what we talked about there? Yeah, excellent. Autocrine, we talked about that, right? Paracrine, I think we talked about that too. All making sense? Do all cells respond to all hormones? What did we say about that? No way. Only cells that have what can respond to the hormone? Functional receptor, right? Okay. Mechanism of action, we talked a little bit about this. Water soluble versus lipid soluble. The differences between each. Which one can get through the cell membrane fairly easily? Lipid soluble, so the receptor is going to be inside the cell, intracellular. Water soluble is going to have a membrane-bound receptor. All right. So water soluble, membrane-bound receptor, classic example that we've talked about. So if I ask you any time about what type of receptor it is, if you say GPCR, you're probably right. So here, a lot of hormones work through G-protein coupled receptors, right? And so you guys are experts in G-protein coupled receptors, so you know all this, but I'm going to go over it anyway, right? Hormone binds to the G-protein coupled receptor, which is going to activate the G-protein, and then you're going to get second messengers generated. So what was the first messenger here? What was the first messenger? What bound to the receptor? The hormone. Hormone's the first messenger. Second messenger is within the cell. Right? So here's outside the cell, here's inside the cell. A lot of times, that second messenger amplifies the signal. Okay? And so here, cyclic AMP is just one example of a second messenger. We can also get calcium as a second messenger. Things can happen once these system is set in motion by a hormone binding to a receptor. And that can involve genes being turned on and off, proteins, enzymes being turned on and off, channels opening or closing in response to activation of the receptor. Right? Is that a question? Okay. Mm-hmm. Mm-hmm. No, so the reason it's amplified is because we activate an enzyme, and as long as that enzyme's activated, we're converting ATP to cyclic AMP, and that amplifies the signal, so we get many second messengers for only a little bit of the first messenger in case of the hormone. Same thing happens with calcium. So for a single hormone binding, we could release a lot, we can free up a lot of calcium that was stored within the cell. And so second messengers in general are going to amplify that signal to cause a whole host of effects. For those that partook in the Miraculin study there, all right? So one of the things is that sits on the sweet receptor and blocks the sweet receptor at neutral pH. Now, the sweet things you guys tasted, it didn't have much of an effect. But the sour things that you guys tasted had much greater an effect. That sweet receptor, what type of receptor is it? G protein coupled receptor. And when it was, when you had acidic pH, when it was sour, right? It was an agonist. It was stimulating the receptor. One of the reasons I think that you see a much more robust response is because when you activate G protein coupled receptors, you get this amplification that goes on with the second messengers that you don't see when you just block the receptor using it as Miraculin when it was acting as an antagonist. All right. So does that hopefully jogging your people's memory about G protein coupled receptors? All right. So here, what we're doing is we're having some sort of chemical that's causing this, and it's going through this receptor to communicate to inside of the cell. So it's how can we communicate or send communication from outside of the cell to inside of the cell? If it's water soluble, it goes through a membrane-bound receptor. Okay? All right, lipid soluble. Lipid soluble hormones are steroids. They're synthesized from cholesterol. You're not going to need to be able to draw cholesterol, any of these hormones. The reason I'm showing you the chemical structure is that similar chemical structures have similar chemical characteristics. So here you can see how closely all of these steroid hormones resemble that of cholesterol from which it was derived. So here we have gonadotropins or sex steroids. Here we have glucocorticoids, which includes cortisol. And we also have mineralocorticoids, which are primary ones aldosterone. Okay? All of these are derived from cholesterol and all have similar structure to cholesterol, and therefore have similar chemical characteristics to cholesterol. And what do we know about cholesterol? It's lipid soluble, right? Okay? Where are the receptors for these guys going to be located? Inside a cell, intracellular receptors. All of these are made in the adrenal cortex. So the adrenal glands involved with making steroid hormones. Not all of them, because a lot of our gonadotropins, testosterone, and estrogen and estradiol are also made in reproductive structures, testes and ovaries is a primary maker. Little bits made in the adrenal glands. Right? So here, steroid hormones can easily get inside the cell, bind to the receptor, and then cause a whole host of things to happen, just like what we saw with G protein coupled receptors. A lot of times what ends up happening is when that hormone binds the receptor, it translocates into the nucleus to cause genes to be turned on and off. And so here, when a hormone is activating a cell or it's influencing a cell, it's causing all of these things to happen, which means to reverse this, is it going to be easy and quick? No, it takes a lot of time to reverse all the things that a hormone-activated cell is having the effect of. All right, so we have an intracellular receptor for steroid hormones, lipid-soluble hormones. Okay? All right, so mechanisms that stimulate hormone release. So let's start off with this panel over here. And this is looking at the parathyroid glands that are little buttons inside of thyroid tissue. So there's specialized tissue here. And if you remember back to 231, parathyroid hormone is released in response to low blood calcium levels, right? Low blood calcium levels are going to release parathyroid hormone, And if you remember back to homeostasis, if we have something that's low, right, what type of feedback is involved? We want to reverse it, so that's positive or negative feedback. Negative feedback, right? And so here, what ends up happening is blood calcium's low, we want to raise it. What is that going to mean with what parathyroid hormone does? It is going to try to raise the calcium levels. Now, what kicked off this whole parathyroid hormone release is that there are blood vessels, capillaries, that the parathyroid tissue is in contact, close contact with, so they can sense whether blood calcium levels are low. So it has a sensor and then also can respond. So here, the release of this hormone is coming because there's kind of detection of a particular, chemical or some sort of parameter in the blood, okay, that's triggering the release. In this case, it's low blood calcium levels, right? So that is one mechanism by which hormones can be released, is through direct detection of, whatever's going on in, say, the blood or some fluid or some tissue, right? It directly is going to stimulate hormone release. Any questions on that? Does that make sense? The second one, we kind of seen already with the sympathetic nervous system, where we had sympathetic nerve fibers innervating the adrenal gland. And so these sympathetic nerve fibers secreted a neurotransmitter that is going to cause release of the hormone. And remember, when hormones get released, they're, by definition, released into the bloodstream, right? Okay. And so here, this is happening in the adrenal medulla. So the adrenal gland, if you kind of think about it as kind of this weird-shaped hat, the interior of the hat's the medulla. The part that's outside of it's the cortex. It's similar to what we talked about with the brain. The cortex of the brain, the cerebral cortex, was the outer portion of it, right? So here, the adrenal gland has adrenal cortex, which is kind of the covering, and then inside of it's the medulla. The medulla is part of this sympathetic stimulation of release of these catecholamines, and specifically epinephrine, norepinephrine, right? Because they're released into the bloodstream, okay, we talk about them as part of the endocrine system as well. So here, nervous stimulation can cause hormones to be released. Okay? Does that make sense to folks? Right? So how does this nerve fiber communicate with those cells? Well, think about the paracrine mechanism, right? There has to be a neurotransmitter released to do this. Okay. Here, was there a neurotransmitter involved with the release? No, it was directly detecting low calcium and therefore stimulating parathyroid hormone release. The last one is probably the most common way that hormone secretion is regulated, in that hormones from one gland stimulate hormones released from another gland. So here, one of the reasons we call the pituitary gland the master gland is that it secretes a lot of hormones to stimulate hormone release from other glands of the body. So here, the thyroid hormone coming from the thyroid gland, right? Its release is controlled by a hormone coming from the pituitary gland. So you have a series or a sequence of events. You have a hormone coming from the pituitary gland, and that hormone's got receptors on the thyroid gland, and when that hormone binds to those receptors, it's going to cause those cells to secrete hormone from that thyroid gland, thyroid hormone, right? Okay? So same thing with the adrenal cortex. So here we're not talking about the medulla, that was over here. Here we're talking about the cortex. There's a hormone secreted from the pituitary gland that binds to receptors on the adrenal cortex to cause hormones to be released from the adrenal cortex. So it's a bit like a relay. right? So you got one hormone coming from this tissue that is going to bind to a receptor in this tissue to cause hormones to be released from this tissue and so forth. Okay. Same thing with this gonads, right? We're going to have hormones released from the pituitary gland, which is going to cause sex hormones to be released from particular gonads, testes, and ovaries. So are these three mechanisms making sense? This is the one we're going to spend a lot of time talking about, and you'll see in the lab. All right. So hypothalamus and pituitary. We won't get through all this. We'll just introduce a few features of this. So remember that we have the hypothalamus is up here, and then it leads through the stalk to the pituitary gland. And the pituitary gland kind of has two sections to it. One that's anterior, so we call that the anterior pituitary, and one that's posterior, so we call that the posterior pituitary. Turns out there's only two hormones released from this posterior pituitary gland. All right. There's a lot more coming from the anterior pituitary gland. The posterior pituitary gland, neurons project to it that start off in the hypothalamus. And so here, these neurons are the ones that generate the hormone that gets released into the bloodstream. Okay? Here, the anterior pituitary, it's going to respond to hormones coming from the hypothalamus that's going to cause cells inside of the anterior pituitary to release their hormones into the bloodstream. So it's a bit of that relay situation we just talked about, okay, between the hypothalamus and the anterior pituitary. Posterior pituitary, though, where are these hormones being manufactured? Originally, they're being manufactured here in the hypothalamus. Everybody follow me on that? All right. So hypothalamus, like I said, is physically linked, and there's a blood vessel network called the portal system that connects the anterior pituitary to the hypothalamus. And then here, the posterior pituitary, we actually have neurons that project into the posterior pituitary from the hypothalamus. Right? And so here, this portal system is kind of like a little bit of a mini highway between the hypothalamus and the anterior pituitary, so that hormones released from the anterior pituitary can make it to the anterior, or from the hypothalamus to the anterior pituitary, so that the anterior pituitary can release its hormones into a bloodstream so they can be systemically distributed. Okay? So here, this close relation between the hypothalamus and the pituitary, we can call this the hypothalamic pituitary axis. So anytime we have two or more glands set up like this in the endocrine system, they're referred to as axes, or an axis singular. Okay? So here we can call this the hypothalamic pituitary axis. If we want to be more specific, we can call it the hypothalamic anterior pituitary axis. All right? And so forth. Okay. So I think we'll leave it there. All right? And we'll pick up with talking about axes and the rest of the hormones on Monday. All right. So today in lab, I strongly encourage people to take some time with looking at these hormones so that you're not overwhelmed with trying to study for the exam between Monday and Wednesday. All right.